So we started new chapter which was chromosome and DNA. Then the second video for us for chromosome. Detail of chromosome today is detail of DNA. We'll cover out everything here. So DNA, of course, we know that's a deoxyribonucleic acid. Right? Then DNA, which is also double helix structure. When we say double helix, this means they have two strands, right? So what else? What we'll see, the DNA was coined, I mean the DNA structure and everything, the name, this was coined by, or the first time it was seen by Watson and Crick. These both scientists saw the DNA and they just told that this DNA is simply a polymer of nucleotides. Now when comes polymer is simply a chain. If this the one part, this means this one part called as a monomer, then this is going to combine so many monomers together, then whatever structure would be formed, that is called as a polymer, right? So what does Watson and Crick said? They says the DNA is a polymer of what? Polymer of nucleotides. DNA is a polymer of nucleoside, but at the time it was quite unknown what is nucleotide. This means they don't know how nucleotide is arranged, what are these things. So this means this entire process, but they were right. Yes, DNA is a polymer made up of nucleotide. Then this nucleotide have again so many parts. This nucleotide also called as a monomer, according to Watson and Crick. But at the time, no one was known what is nucleotide, so this entire process remained as a hypothesis. Hypothesis, something which is not true, not acceptable at that moment. But yes, then later on there was another scientist, few more talks and something else we'll discuss. So this means till here no DNA was formed, right? So at the time everything was done, like nothing was. Then another scientist came, and that even not another, but this these scientists again start researching on DNA. What is exactly DNA and their types? Like how is it that formed? So they saw that DNA is a polynucleotide. Poly means many or different. So even before that, they saw that DNA is a polymer. They were like confirmed DNA is a polymer. But when they were asked what would be the monomer, then so at the time they replied monomer is nucleotide, right? And yes, it was right. Today we accept it, right? So this means DNA, then it was said that DNA is a polynucleotide. This means if this the one nucleotide and its completely chain is going to be combining, then that's a structure called as poly. Of course we know that means many, and this is nucleotide. So many nucleotide combine, they form a structure that's called as a polynucleotide. Again, there comes the division. Division like what? This nucleotide is again divided and made up of three different things. What are these? First one called as nitrogenous bases. What were these? Nitrogenous bases, which are simply organic compounds. Organic compound, this means they will have carbon and nitrogen hydrogen rings, right? Then this organic compounds were again divided into two parts. First one called as purines, second one called as pyrimidines. In pyrimidines only single ring was formed. Here two rings, this means in the middle they will have two rings, but here in pyrimidines there will be only one, they were told. So here this means double ring would be there. So example was adenine and guanine. When comes the example of pyrimidines, this means there will be single structure. So there was the example of thymine and cytosine. When comes RNA, said thymine is not present in RNA, so there would be replacement with the uracil. So this means till now nitrogenous bases were very much clear, right? So I need to erase this so that I could manage it out rest. Then the second, what was second? Second, it was said that the nucleotide is made up of nitrogenous bases. Secondly, pentose sugar. Now when we talk about the pentose sugar, what is this? Pentose in mathematics and all these terms we have studied pent, hex, hept. Pent means 5, so this means 5 carbon ring structure would be called as pentose sugar. Sugar, if we are discussing sugar, wherever you just see word sugar, this means we are discussing something related to carbohydrates, right? Because in carbohydrate we study sugar, glucose, all carbohydrate would be called as sugar, but here there would be a 5 carbon like structure. So if I just make it out, let's go with another color now. This is structure. Here we'll see all these nitrogenous bases, 1 2, like first suppose if I'm taking adenine, 5 nitrogen spaces are present, right? So I hope this will be only a simple structure. Pentose sugar is present, nitrogen basis, pentose sugar, and the lastly we have one more term. Lastly we have third one, that's called as a nucleoside. Not nucleoside but the phosphate group. Phosphate group. So what is that now? When we discover the phosphate group, this means phosphate group is simply nucleoside. Before when I discuss that, here I'm just writing nucleoside plus phosphate. When they both will be combined, they form a structure called as a nucleotide, right? Nucleotide. Even when we also said if there is nucleotide, this means there's the, we are discussing nucleotide. Nucleotide is made up of nucleoside plus phosphate group. When they will combine, they will form this structure. So I hope it will be clear. Then comes the phosphate group. This means this simply phosphate group. Will addition of phosphate will be there. So when we'll add phosphate, this mean addition of phosphate. So till now, like everything should be very much clear. This is the nucleotide structure, and nucleotide was what? It was a monomer of DNA. So I hope it will be like very much clear. Then what comes next is about, so here we have other things which is about nucleotide. We are just grouping or like combining them. So what I'm exactly doing is about we're adding phosphate plus sugar plus any nitrogenous bases would be form a structure. Now what exactly is this? This means if we add phosphate plus sugar plus adenine, right? If I'm adding adenine, what they'll form? They'll form adenylate acid. Second, which comes is about phosphate plus sugar plus cytosine, right? So what will it form? It will form cytoclic acid. This means we are forming acid by these all nitrogenous bases. Then phosphate plus sugar plus guanine. It will form what? It will form guanylate acid, right? Then comes next one, which is about thymidine, phosphate plus sugar plus thymine, which will form thymidinic acid. In the end, even this thymine, of course, this is, I don't know how many times I've repeated, thymine, which is only present in DNA. So when we talk about the RNA, we know there's something different. So in RNA, phosphate plus sugar plus uracil would be added. Then what it will form? It will form uridylic acid, right? So I hope it will be clear. Then comes another term, another important MCQ. DNA is what? Heteropolymer or homopolymer? So basically this is heteropolymer. Hetero means different, so different nitrogen bases will be combined and they will form a structure called as heteropolymer and nucleotide, these all things. So I hope entire detailed video for DNA would be very much clear. Thank you for watching. Bye bye.